The type 1 interferon system provides a powerful defense against viral infections. The cellular recognition of virus by intracellular receptors like RECOMB can lead to the activation of the key transcription factors of interferon, the nuclear factor kappa B, and the interferon regulatory factors 3 and 7. The nuclear translocation of these key transcription factors can in turn induce the expression of type 1 interferons, like interferon beta. The uptake of interferon induced the STAT signaling pathway in an autocrine or paracrine manner. Activated STAT may induce the expression of interferon-stimulated genes, which in turn trigger an antiviral protective state in the cell. To examine interferon expression, we used fluorescent reporter cells which stably expressing interferon beta turbo GFP. After infection, the reporter cells showed an extremely heterogeneous interferon expression, where the single cells started at very different time points to express interferon. The onset of interferon expression varied in individual cells between 5 and 20 hours after infection. During our study, we tried to work out several questions. First, what determines heterogeneous interferon expression? Is it the virus or the host cell? Is the observed stochastic expression a special feature of the interferon beta gene? And how can a reliable antiviral protection be achieved? To quantitatively determine interferon expression, we jointly measured viral protein and interferon expression by flow cytometry. The FACS data demonstrate that viral replication, here the white panels, are necessary to induce interferon, upper panels, but it's not sufficient. Okay, normally you shouldn't see the dots here. I don't know what happened. Okay, so, but a large number of cells, namely 2/3 of the infected cells, didn't express interferon. And these cells which expressed interferon showed a strong range in intensity. By time-lapse microscopy, we observed the onset of interferon expression after infection with different virus concentrations. The interferon-producing cells started at very different time points to express interferon. To analyze if this heterogeneous interferon expression is due to different time points of viral infection or reflects host cell intrinsic noise, we bypassed viral infection and transfected the cells with double-stranded RNA analog poly-IC. Also after transfection with different poly-IC concentrations, we observed an extremely heterogeneous expression of interferon. And this variability was quantitatively comparable to the viral infection case, as seen by the same order of magnitude of the coefficients of variation. To analyze mechanistically how the cell-to-cell heterogeneity in individual cells arises, we had a closer look on the interferon induction pathway. The nuclear virus sensing by intracellular receptors like RECOMB can activate the key transcription factors NF-kappa B and IL-7. We monitored these both key transcription factors in dual reporter cells by live cell microscopy. After infection, we observed that the nuclear translocation of both transcription factors happened simultaneously in a single cell. but this joint nuclear translocation time differed between individual cells. And this difference was between 7 and 20 hours post-infection. We also looked to the relation between transcription factor activation and interferon beta gene expression. And here we observed that the majority of the cells, namely 90%, expressed interferon after they have translocated the key transcription factors. And the time interval between transcription factor activation and interferon beta expression varied in individual cells. Therefore, we suggest that the decision to express interferon is primarily taken upstream of the transcription factor activation. The uptake of interferon can induce the expression of interferon target genes, which in turn trigger an antiviral protective state. To analyze the interferon response, which shows IRF7 as a prototypical interferon target gene, we stimulated IRF7 reporter cells with different amounts of interferon. And here we observed a dose-dependent binary pattern of a responding subpopulation, which is protected against viral replication, and a non-responding subpopulation in which virus can replicate. These data demonstrate that also the interferon response is an all-or-nothing switch in individual cells. We established a multi-scale mathematical model which combines the antiviral signaling and interferon induction and the expression of interferon target genes in individual host cells together with the cell-to-cell communication by secreted interferon. To estimate the parameters of our model according to the experimental data, we firstly estimated the parameters for the intracellular viral replication. With these parameters, we calculated the weights for the activation of the key transcription factors and the interferon beta expression by fitting the corresponding switching time distributions. This enables us to explain the dynamics of interferon induction, interferon secretion, and the expression of interferon-stimulated genes. We calibrated our model for a single high-dose infection and simulated low-dose infections where the interferon-expressing cells are sparser. Our model predicts a strong paracrine signal propagation of antiviral protection through interferon with up to 40 times as many interferon responding cells than interferon producing cells. This prediction agreed remarkably with the corresponding measurements. Therefore, we suggest that few cells which express interferon act as sentinels of viral infection to protect a large number of cells surrounding the infection site. Let me summarize by answering our main research questions. The observed heterogeneity in interferon expression is not controlled by virus, but is primarily due to host cell intrinsic noise. Our study of the interferon response demonstrates that also interferon target genes operate as stochastic switches. And mathematical modeling combined with experimental validation demonstrate that a reliable antiviral protection in the context of this multi-layered cellular stochasticity can be achieved by paracrine response propagation. I would like to end by thanking our collaboration partners at the Helmholtz Center of Infection Research in Braunschweig, Germany, Ulfert Renz, Mario Köstet, Hans-Jörg Hauser. I thank my colleagues at the German Cancer Research Center, Michael Floßdorf and Christoph Kali-Coulet. A special thanks goes to my supervisor, Professor Thomas Höfer, and you for your attention. Can you speak up a little bit? Sorry. Yeah. It was we observed the interferon expression with no, we just observed oscillations for the key transcription factors. They are really oscillating when they go into the nucleus.
Okay, thank you.